turn off your video soon after we start the talks. So to avoid any disturbance. So that is one. If you have any questions to be asked, please type in the chat box. Then the chair will read out the questions to the speaker. And others, as far as possible, please do not barge in because it creates unnecessary disturbance. So with this, now today I have requested Sulakshana to take the chair and conduct the proceedings. Sulakshana, is over to you.
Thank you, Professor Surya Prakash. Okay, today we have two speakers, and the first speaker is Professor Neeraj Sinha from CBMR Lucknow. Professor Neeraj Sinha received his bachelor's degree from Banaras Hindu University and his master's from Jawaharlal Nehru University, Delhi. He earned his PhD from IISc Bangalore. Subsequently, he moved to US for his postdoctoral research at UCSD and Iowa State University. After his postdoc, he joined CBMR Lucknow, and currently at CBMR, he is using various solid-state NMR techniques to understand the molecular pathways behind various diseases. He was conferred with Professor S. Subramanian's 60th birthday lecture award in the year 2015. With that, I request Professor Neeraj Sinha to proceed with his talk.
Thank you, Sulakshana. And so can you see my screen, the screen?
Yes, we can see.
Yes. Okay. Yeah, so thank you, Sulakshana, and thank you, organizer, for giving me opportunity to present some of our recent results on the solid state NMR of bones and cartilage. We have been trying to understand the bone structure by applying some new techniques of solid state NMR for quite some time. And and recently we have got some new results, which I would like to share in this platform. But before we begin, I would like to dedicate this talk to one of my old friend who is no longer with us, Professor Atreya. And this is one of the pictures which we took during our PhD days. And we immediately became friends, and it was a friendship for the lifelong. And he was a very good. He was a very good friend and a colleague. And sadly, he is no longer with us. So I have divided my talk as first introduction to bones and cartilage, and then we could detect some lipid inside the bone matrix, and then some results on the biosolid cryoprobe. Recently, cryoprobe for the solid has been developed, and we could try some of our bone experiments on the cryoprobe, and we got some interesting results, which I will try to present. And finally, I will try to apply DNP enhancement for bones and cartilage and what it can give as a bone structure and cartilage structure. So bones are very important component of human neuromuscular skeletons, and it not only give us shape as a human body, but it also support for heart, lungs, and bone marrow. And it is act as a protection for brain, uterus, and other internal organs in a human body. And not only that, it also allows for attachment for muscles, for movement of limbs. And so these are just the structural part of the bone, but bone has a lot of biological role to play in a human system. For example, like it act as a mineral reservoir for calcium and phosphorus. So whenever a human body need calcium or phosphorus, then bone is the source for that. Not only that, but it also provide a defense against medical condition, acidosis, like when the pH of the blood changes, then the bone is the source for controlling the pH. and it also acts as a trap of dangerous material in human body such as lead. So keeping in view that bone is a very important human system, it is important to understand various bone related diseases. Cartilage, on the other hand, is a slightly flexible and smooth connecting tissue, and it is mostly there in between the joints. And due to ECM, cartilage allow the tissue to bear mechanical stress. So whenever there is a mechanical stress, then cartilage provide a cushion for that. And in a respiratory tract, ear, nose, and cartilage form a framework for supporting the soft tissues. And sometimes due to disease condition, the cartilage degradation happens, and this is known as osteoarthritis, which is a very big problem for old age people. So, and it is basically because of some damage which happens in the cartilage structure. So with solid state NMR, we can try to understand some of the structure and how these damages takes place in bones and cartilage, and what we can understand about the mechanical property of those bones. So bones are also a living system, like it has a blood vessels coming from the part of that, and this is the part from the compact bone, and this is the spongy part of the bone which is outside the bone surface, and it also has a lymphatic vessels where the blood flows, and then bone regeneration happens. So sometimes because of some accidents, the bone fracture happens. And since bones are living system, we don't have to do anything. Just we have to make sure that bone is not moving. And then because of its blood structure, the bone healing happens. And these are the different stages of bone fracture healing, which automatically allow bone to regenerate it during the fracture times. And then it gets healed up, and then the fracture gets repaired. In a younger people, the bone healing is fast, whereas for the older people, the healing is very slow, and sometimes it takes a long time. So this medical condition is known as osteoporosis. So in terms of different component and what we can understand about the bone structure. So bone is basically a multi-scale hierarchical system where if we keep on going in a different uh, like a scale from 10 millimeter to all the way up to 10 to the power 6 micrometer, then on different scale it has a different kinds of structure. For example, like in this region, it is mainly the structure has a framework kind of thing, but if we go deeper into 100 to 200 micrometer, then there are holes, and then the canals are there. And then if we can go even deeper to the micron level, and then to the nanometer level, then there are collagen fibrils inside the bone structure. And on top of those collagen fibrils, there are hydroxyapatite crystals. And these are the mineral crystals, inorganic mineral. And then the collagen provides a support or framework for the bone. So, and then the collagen structure can further be simplified in a 1.5 nanometer range. Then it has a triple helical kind of structure. So due to some disease condition, many times the bone mechanical property gets compromised, like in osteoporosis. So since it has a hierarchical structure, we can try to understand what happens during this time, which gives rise to different types of mechanical property at the higher level. So solid state NMR is one such technique where we can probe the structure of these different components in this level in order to understand what is the factor which give rise to the mechanical properties of the bone at the higher level. So in terms of components of bone, it has mainly inorganic minerals, which act like a tiny crystal at the top of this collagen fiber. And there are organic proteins, like collagen is one such protein, and then there are certain other proteins. And then there are lipids molecule inside the bone, and the one very important component of bone is water molecules. So bone also has a lot of water component inside the bone, and it is the interaction between all these components give rise to bone, a unique mechanical property which also keeps on changing throughout the life and regeneration. So for osteoporosis kind of disease condition, BMD, bone mineral density, was supposed to be a very good indicator for bone mechanical property, but recently the understanding has been changing, and people believe that the water plays a very crucial role in bone in terms of deciding its mechanical property, like stress bearing and fracture conditions and fracture propagation. And all these things can be seen, like here, this is the bound water measured in a human bone for younger people and older people, and pore water content measured for younger and older people. So it turns out that as the age progresses, the bound water and pore water content significantly changes inside the bone matrix. Not only that, the peak stress and the peak stress is also a function of bound water fraction and pore water fraction. So all these results shows that even though the BMD of the per particular person can be very high, but still because of the depletion in the water content, the bone mechanical properties can be very different. And all these results started like how the water plays an important role inside the bone matrix, which we can easily probe by solid state NMR experiment. So one such thing that we have been trying to do is applying this solid state NMR lens to look at the bone microstructure, and then whatever we get is giving the understanding of how these different components of the bone are arranged inside this. So one such result is about the lipids. So on the left hand side, we have the proton NMR spectrum of the bone. So you can see that it's a very broad component with lipid resonances coming somewhere here in a sharper part. And this proton spectrum can be fitted with the mobile water component, which can be a slightly sharper, and the bound water component. Okay, so good thing is that this lipid have a longer relaxation time, and then we can do CPMG kind of experiment to selectively filter out the mobile and bound water content and observe the lipid resonances. So if we do a proton CPMG kind of experiment and choose a T2 filter in such a way, then we can get a sharp resonances coming from the lipid part, although in bone everything is in the intact form. And these lipid resonances can be assigned, and these are mainly triglyceride resonances, and then we can assign almost all the resonances which comes from the triglyceride. So what we did was, like, we created three conditions where we have the bone which is fully hydrated, and then we exchange proton with the deuterium by HD exchange, and then we dried the bone. So these are the three NMR spectrum of the bone in three different conditions where we have changed the water content inside the bone matrix. So we can see that the lipid resonances are mostly unaffected. And then by using the CPMG experiment, we can observe what is the lipid resonances happening into that. Not only that, we can measure the intensity of the different lipid resonances for the CPMG delay time, and can measure how the relaxation is happening, and we can measure the T2 value. So it is best fitted with a bi-exponential, where we get a fast T2 component and a slow T2 component of the bone matrix. And we could do this for almost all the resonances of the triglyceride, and then we can terminal CH2, beta CH2 coming from this triglyceride, and we could measure that. So since we can measure the fast decaying component and slow decaying component, we went ahead and tried to measure what will happen to this decay constant when we change the water matrix inside the bone, like we do the dehydration or we do the HD exchange. So this is one such result where we have the slow decaying T2 component as well as the fast decaying T2 component for bone condition where we have a hydrated native bone where the full water content was there, and then when we have a HD exchange or dehydrated, and we could measure the change in the lipid resonances T2 as a function of CPMG decay constant. It turns out that as the bone gets dehydrated, T2 increases, but this increase or water dependence is not identical for almost all of that resonances from the triglyceride. It is mainly affecting the two part. One is coming from this part and this part. So this shows that these are the part where the water is strongly interacting with the lipids inside the bone matrix. And all these things can be probed because we could measure the decay constant at 3 different conditions and compare between the two. So the part where the water is strongly interacting shows the maximum change happening because of in the decay constant. So this was a proton part of that, and we can also measure the carbon-13 spectra, which will mainly come from the collagen part of that of the bone matrix. And collagen is the main structural component of extracellular matrix, and it has a triple helix structure. And collagen is a very compact triple helical structure with the sequence like glycine XY, where X and Y are mostly proline and hydroxyproline. These are very small amino acids, and in terms of size, they are very small, and the collagen structure is very compact and very rigid. But in X and Y, there can be some other amino acids which can be slightly bigger. For example, like these amino acids and their occurrence in different types of collagen are given here, and they are in very less concentration, less than 0.5%. So, but these amino acids have a bigger size. So it is always a question like how those bigger amino acids play a role inside the collagen triple helix structure, because they are quite bulky and some are charged, some are aromatic amino acids. So it is a open problem like what are the interaction which stabilize these amino acids inside the collagen matrix in a native system. So solid state NMR can provide some information about how those amino acids are there because we can measure them. So, but the problem with all those experiment is like number one, the concentration of organic part is in bone is very less. So that gives that, and also the carbon is in natural abundance. So it has a problem like, although it can map the subtle structural changes in bone structure, but sensitivity is one such limitation to apply this experiment in bones and cartilage. So good thing is like the recent development in biosolid cryoprobe has increased the sensitivity, and then dynamic nuclear polarization can also enhance the sensitivity of solid state NMR experiments. So we have been trying to apply these two techniques to in the bone structure. Not only that, people have been trying some other techniques the way back in 2014. One is like doping the bone matrix with a relaxation agent, gadolinium DTPA, which reduces the relaxation time, and then we can pulse faster and acquire more signal, and then ultimately we get a sensitivity enhancement which can give about 2 to 3.5 times reduction in signal averaging time. One interesting approach by a group from Cambridge uses heavy mouse, where they label the C13 and N15 of the bone in the entire mouse. So it's a very expensive technique, but it gives a very interesting information about the bone matrix and its collagen structure inside the bone matrix. And then we have been applying the dynamic nuclear polarization to bone matrix, and then we can see these experiments. So in terms of biosolid cryoprobe, this is a fantastic probe where we can get approximately 4 times signal enhancement. So this is the conventional room temperature DVT probe from Bruker, and with the same signal to noise ratio and the number of scan, we can get approximately 4 times signal enhancement in a natural abundance C13 spectra of the bone. And this allowed us to do some more experiment to measure interactions inside the bone matrix of those systems. So natural abundance C13 spectra of bone can be acquired very easily, and then most of the resonances can be assigned. And interesting part is like we are getting arginine, C delta, and ring carbon of the aromatic amino acid inside the collagen. So its concentration or its occurrence is less than 0.5%, and we could get resonances coming from there. Apart from that, we can get maximum resonances coming from different parts of the collagen like hydroxyproline gamma and glycine alpha and proline, hydroxyproline part of that. And another interesting resonance comes from the citrate, which comes around 78 ppm. And citrate is an important molecule, and it plays a very important role in the bone structure, because that is the molecule which club the inorganic minerals with the collagen protein. So this is the resonance which is the closest from that. And we can also get N15 resonances in a natural abundance from the bone spectrum by applying this biosolid probe in a reasonable time. And then, so these are mostly the resonances coming from the backbone part, and these are the side chain resonances coming from the side chain of the N15 inside the bone matrix. So since there is a sensitivity enhancement, we measured the short and long range correlation by head core kind of experiment. And if we use a short contact time, we get a 1 bond correlation. If we use a long contact time, we can get a long correlation, more than 1 bond through space correlation inside those matrix. And some of those correlations can be carefully looked into that. And these are the correlation representing intrahelix hydrogen bonding network coming from glycine, proline, and hydroxyproline residue, and charge pair interactions, as well as NH-pi interactions, since we can assign most of these resonances here. So the other interhelix hydrogen bonding network can also be measured from these long-range correlation, mainly phenylproline, hydroxyproline, CH-pi interaction, and hydroxyproline-proline hydrophobic interactions. All these interactions can be measured because we can get a long-range correlation. So such interactions are not possible to measure in a native system without these sensitivity enhancement and these things. So these are the hidden interactions which cryosolid probe, cryoprobe has made us to observe inside the matrix. So in the last, I will talk about DNP enhancement. So this is natural abundance C13 spectrum with the microwave on and off, and we could get approximately 30 times signal enhancement or more. And this is the natural abundance C13 spectra with the NP enhancement for bone as well as for the cartilage. So cartilage has a less concentration for the collagen, and it has a different types of collagen, but most of these resonances can be observed. And not only that, in the bone matrix, we could observe the aromatic amino acids resonances, as well as the citrate resonances and hydroxyproline gamma resonances. And in the cartilage, we could observe the GAX resonances, GAGS, which is an important component of the cartilage extracellular matrix. And both of these resonances can be observed by DNP enhancement. So we went ahead and then looked at the head core experiment, and to see what kind of interactions we can see in the 1H C13 head core experiment, especially about the aromatic amino acids and its interaction with citrate and other part of that, and aromatic amino acids and citrate with charge pair interactions, mainly coming from arginine C beta, coming from this path, and lysine C beta and glutamine C gamma. So we can also look at the cartilage part
Yes.
where we
Hello.
can, hello. Yeah, we can all,
Okay, hello.
yeah, we can also look at cartilage part where we can see the interactions of different parts of the gags and how it is interacting with the collagen inside that, mainly gags with the collagen residue coming from hydroxyproline gamma and
Yes.
arginine alpha.
Hello, my name is Jonathan from Microsoft Tech Support. How may I help you today?
So this is our result with a natural abundance 13C
Sir,
double
I
quantum
see hearts
single
all over
quantum
the screen. There's
correlation.
hearts. Do you see this? Hearts? Why someone put the hearts? He's drawing on the screen.
uh, with the DNP enhancement of bone and cartilage. I don't know how it got there. Should I continue or?
Yes, we go ahead. I think there's someone who's creating some trouble. I've removed the person. Yeah.
Okay. So yeah, so this is a natural abundance 2D 13C double quantum to single quantum correlation experiment in a natural abundance, and this has been made possible by SPC 5 coined as sequence where and this was done under the DNP enhancement, and this left side is the result for the bone is extracellular matrix, and this is the cartilage one, and we can get short range one bond and long range correlations, which is more than 5 angstrom. And mainly the aromatic amino acids correlation can be observed in this kind of system. So with the DNP enhancement, we could also observe backbone natural abundance N15 spectrum of bone and cartilage. And this allowed us to look at the correlation of N15 with proton. So mainly the backbone correlation and the side chain correlation, and how it map when the collagen is inside the bone matrix or inside the cartilage, which has a more water and other component is there. So with this, we can conclude that water-dependent subtle structural changes, whether it's a bone, extracellular matrix, or cartilage, can be mapped by solid-state NMR. And not only that, we could look at the lipid part, and how the lipid is interacting with water can be probed by lipid dynamics. And cryoprobe gives a very good boost in terms of sensitivity, and many of these experiments can be carried out in cryosolid cryoprobe. And of course, DNP open up a new avenue to probe these interaction in the native state. So with this, I would like to acknowledge my research group who did many of these experiments, and funding was from the Department of Biotechnology, UP government, and DST. And for DNP studies, we collaborated with National High Field Magnet Laboratory. And of course, biosolid cryoprobe experiment was done at Fallendon, Switzerland. And thank you so much for your attention. And if there is any question, I would be happy to take that.
Thanks, Neeraj, for such a wonderful talk. You have many questions here. The first question is, do you take intact bone or make a powder while making the samples?
Yes, that's a very good question. Our aim is to make sure that the bone structure does not change as we do solid state NMR experiment, and water is a very critical part of that. So we have done some experiment where we took an intact bone, and then the water was preserved for a long time, and then the matrix, everything was intact. So initially we used to do intact bone experiment. Now we have found that if we make a small flakes of the bone, then we can do very good magic angle spinning, and the water content will also be the same. So we don't make a powder, fine powder. We make a flakes of those bones and then do magic angle spinning on that.
So the next question is, could you please comment on how do you prepare bone samples for SSN? It's the same question. So I think you make flakes and yeah.
Yes, yes, and the flake should not be a very small. Then the water will go away during the magic angle spinning, and then it will no longer be the
So it's
thing.
a particular size, sort of. So how do you make those flakes?
By knife, we just cut a small pieces into that, and then it comes out from that.
So does lipid composition and concentration changes in bone of different organism? Does lipid changes the composition of and concentration? Huh.
Yes, yes. So lipid mainly comes from the cell part of that. So if the bone is near the bone marrow part, then it will have a very high lipid component. And if it is from the outer part, then the lipid concentration will be less. This is something which we have observed. Like if we take a different types of bone, then the lipid will definitely be different. But in this study, we were mainly interested in looking at the lipid, how it interact with the water. So different types of bones will have a different lipid component into that. That's correct.
And can you comment on the amino acid composition variation in collagen protein in different organisms?
As far as the bone of a mammalians are concerned, the collagen components are same. But different other types of organelles will have a different distribution of amino acids in the collagen. But for mammals, it is mostly same. That is why we could take a goat bone and do the experiment, or cow bone or any bone into that. Mammals have the similar kind of structure.
So I think, okay. What was the time required to acquire the natural abundance 2D head core experiment on the biosolids cryoprobe? And is it 13C proton detected experiment?
Yes, it was a proton detected 13C experiment, and we could do in less than 24 hours for the C13 proton, and good correlation. And for N15, it was not more than 2 days.
I think there is some distraction. Vivek and Jeet, can you please look into it? It's like a lot of distraction coming. Jeet and Vivek, please, can you look into why the distractions are coming? I think somebody is playing some mischief here.
I know, we are working on that. I don't know, this is very ridiculous. I have removed 4 or 5 participants till now, but yeah.
Someone called Maggie, and now someone called Maggie just screamed, I think. Yeah.
Okay.
Okay. Okay. So one of the audiences wants to know more about the specific water localization in lipids. Your arguments on that, like, he's confused.
Water localization in lipid.
Yes.
Sure.
Yeah, so actually it is, I do not get your arguments on specific water localization in lipids. That's the...
Yeah, actually what we observe is that there are certain resonances in the lipid which are very much critically dependent on the water content. For example, like if you change the water content, then its relaxation time changes drastically. Whereas for others, the change is very less. So based on this result, we concluded that those are the part where the water is interacting heavily with the lipid part.
Could you also see hydroxyl lysine?
Hydroxyl lysine, we have not seen any such resonances on that collagen.
How do you ensure or maintain hydration content in bone during experiment?
We keep on measuring the proton NMR. Proton NMR itself will tell us whether how much is the free water, how much is the bound water. If by starting the experiment we have a proton NMR, and at the end of the experiment it is exactly matching, so we make sure that the water content has not changed during the experiment, and this is very critical also.
So with that, I would like to thank Professor Neeraj Sinha once again. And now we will move on to the next talk by Dr. Haribabu Arthanari. Dr. Hari received his bachelor's in chemistry from the Madras Christian College and his master's in chemistry from the Indian Institute of Technology, Madras. He did his graduate studies at Wesleyan University with Philip Bolton and earned his PhD in 2004. He then joined the laboratory of Gerard Wagner for his postdoctoral training. He was promoted to a lecturer in 2010 and moved to his independent position in 2016. Dr. Hari is now assistant professor of biological chemistry and molecular pharmacology at Harvard Medical Center. His group utilizes a combination of techniques, including NMR spectroscopy, high throughput screening, and biophysical and cell-based assays to map hotspots in the protein-protein interaction interface. So with this, I would request Professor Dr. Hari to continue with his talk.
Thanks.
Hari, can you share your
unmute?
screen? Yeah.
Sure. Let me share my screen and see if this problem goes away. Do you guys see any of the hearts or anything like that? Or
No, no.
Oh.
Very
Not
good.
in
So,
your screen.
thanks, Lakshana, for that lovely introduction. And it's a bittersweet moment to present at this August gathering, especially when Atreya is not there. For the last 10 years of my visit to India, the one thing that was constant is a trip to IISc to meet up with Atreya. And in addition to being a phenomenal scientist, as he is, he's just an awesome human being. He's always kind, very generous with his time and ideas, and harbors the curiosity of a child. This is actually one of the photos I chose from one of his students. And for people who know Atreya, you can see that look whenever you tell him an idea, either good or bad. He has this, his eyes just lights up. And he harbored almost a Da Vincian spirit to his research. There are nothing that put boundaries to what he can think about. He could think about MRI, he could think about metabolomics or cancer biology with an equal spirit. And a window to Atreya's brain could be seen in his bookshelf. And if anybody has visited his office, his bookshelf carries books from pure math to physics to engineering to philosophy to architecture and mythology. He was a scientist where the science didn't have any borders, and he was a person where his heart didn't have any walls, both figuratively and metaphorically. And today we all miss him, and I miss him a lot. And I think the best way to remember him is to inherit his sense of curiosity to science. With that, I'd like to talk to you guys today about some of the NMR methods that we've been serendipitously thinking about or developing to address spectroscopy of large systems. Now, just to set the scene, before that, I'd like to recognize the people who actually did the work that I'm going to present. The work is predominantly done by 3 different people: Abhinav Dubey, Paul Coote, and Scott Robson. And Abhinav is from Atreya's lab. Now, to set the scene, for most of us in the solution state NMR spectroscopy, we use this workhorse experiment, the heteronuclear single quantum coherence, or the HSQC experiment, as a starting and an end point for most of our investigations. And be it either interactions, studies, or looking at relaxation, dispersion, looking at things like exchange using CEST and DEST experiments, this HSQC becomes a starting point for us. However, the bottleneck in using this HSQC experiment is our ability to correlate each one of those resonances to a resonance in the primary sequence by a process we call resonance assignment. And this bottleneck becomes complicated for when you go to larger protein systems. When you go to larger protein systems, actually before that, the way we arrive at these resonance assignment is by using something called the triple resonance experiment, where we transfer magnetization along the protein backbone. And we use these correlations then to back correlate each one of this peak to an amino acid in the primary sequence. But when we go to larger proteins, there are two specific problems that arises. One of them is the complexity of the spectra. Here I show you two different spectra. One is an 8 kilodalton protein, and another one is from an 80 kilodalton protein. And as you can see, with the larger system, there's proportionally more number of resonances. And this leads to overlap in the spectral space. And I show this in a 2D HSQC. And within the context of a triple resonance experiment, what you'll see is this thing called chemical shift degeneracy. Here I show you a number of strips from an HNCA experiment, and I'm looking for a match for tyrosine 190. And you can see there are about 3 different strips that has the same exact chemical shift I'm looking for. And larger the protein, the more the chances you're going to see this degeneracy problem. And in order to alleviate this problem for smaller proteins, what we do is that we look for additional dimension of matches. For example, we record an experiment like the HNCA-CB or the HNCA-CO, where if there is a degeneracy in the CA chemical shift, we're hoping that the CB beta or the carbonyl chemical shift can break this degeneracy. However, for larger protein systems, we encounter the second problem, which is relaxation. So in general, and mostly in solution NMR spectroscopy, one transfers magnetization between nucleuses using one of 2 different approaches, either a scalar coupling or a dipolar coupling. And the latter being manifested itself is in a Nosies and Rosie experiment, but predominantly we use scalar coupling to transfer magnetization. Now, within the context of the protein backbone experiments, what you would see is that the transfer, the coupling constants which we use to transfer is actually rather weak, except for the NH, which is about 90 hertz, and the C alpha CO is about 55 hertz. The rest are in their teens. Now, as a result of that, what's going to happen is that you need to wait for 1 over 2J time period for this transfer to happen. And during that time period, relaxation hampers your ability to transfer this in larger systems. Plus, we also depend on out and back strategies. Essentially, we like to start with proton magnetization and end with proton magnetization, as shown here in an HNCA pulse sequence, because our inherent sensitivity of an NMR experiment is related to the gamma of the excited nucleus and the gamma of the detected nucleus to the power of 3/2. So it's rather beneficial for us if we start with the proton magnetization and end with the proton magnetization. On the flip side, this leads to additional transfers, which also cause relaxation. An analogy I'd like to give is these two different phones, which most of us are familiar with. And this would be a simple HSQC experiment, and this would be one of those backbone triple resonance experiment, which has additional apps or additional transfers. But as a result of that, the net magnetization when you're detecting is very low. Now, for example, here is an old a figure from an old paper by Michael Sattler, and you can see the relative sensitivity of the other backbone experiments, the HNCA-CB and the HNCA-CO, with respect to the HNCA, which provides sequential connectivity. That's about 13% compared to an HSQC. And here I show the same thing using another example where I show you a TROSY HSQC of a 38 kilodalton phosphatase, and here is a projection along the carbon dimension of an HNCA experiment. Now you can see that you see about a large fraction of this expected peaks. But here's an HNCA-CB for the same protein that was recorded for twice as long, and you see only 12 correlations. So here's a problem that we have. For larger proteins, there are 2 different problems. One is the degeneracy that comes from a lot of peaks, and the second is the relaxation that happens, which makes pretty much HNCA as the only experiment that gives you anything that's useful that has both correlation to the N and the N-1 that allows you to do sequential assignments. Though in theory, you can assign an entire protein just based on HNCA, the C alpha chemical shifts, here I show you the chemical shifts for the C alpha in blue and C beta in red across different amino acids from the BMRB database. But you can see the C alpha chemical shifts are rather degenerate. I mean, they're all around 55 ppm when compared to the C beta, where you see some differences between different amino acids. And this essentially adds to the degeneracy problem, causing severe overlap. So the simple question we asked ourselves is that, can we alleviate this degeneracy problem by extending the resolution into the C alpha dimension? So normally, when we acquire a spectrum in an HNCA, your resolution in the C alpha dimension is about 40 hertz. That's a spectrum that's shown here in cyan. We want to get the C alpha resolution to within 5 hertz, and that's the spectrum that we've shown in blue over here. Now, when this is extended to such a high resolution, now the degeneracy problems can be partly alleviated. So this was a very simple idea that we started off with. Now the question is that, how do we get more resolution? It's pretty simple. You just record the FID for a longer period of time. Here I show you 3 different FIDs with increasing acquisition times. And as you can see that going from top to bottom, if you increase the acquisition time, your resolution increases. But this is also subjected to the laws of relaxation. If during this acquisition time relaxation happens, then you can see that it doesn't matter how long you acquire, there's no increase in resolution. So the simple thing to keep in mind is that we can extend the resolution by acquiring longer. And here acquiring longer is in the indirect dimension because that's where you encode the carbon chemical shift. And if relaxation allows, you can get good resolution. Now the question is that, does relaxation allow that? Here I show you a plot of relaxation rates on the y-axis with the molecular correlation time, or simply molecular weight, on the x-axis. And here you'll see 2 different lines. One is a dotted line, other is a solid line. The dotted line refers to the relaxation at 500 megahertz, and the solid line refers to the relaxation at 800 megahertz. Now, what I want to grab your attention is to the relaxation of the C alpha when it's protonated, to the relaxation of the C alpha when it's deuterated. As you can see, the C alpha relaxes extremely slowly when it's deuterated compared to when it's protonated. Actually, it relaxes slower than the TROSY component of your amide proton. The only thing that relaxes slower than the C alpha is your nitrogen TROSY component. So which means that when your protein is deuterated, you can use the slow relaxation property of C alpha to extend your resolution because you have the time to do that. Now, to put this into perspective, something that's more practical, you approach your line width, which is the best resolution that you could ever get, if you could sample in your indirect dimension or your direct dimension to about pi times T2. So if you're able to sample to pi times T2, even for a protein of about 182 kilodalton, which is the proteasome, you can get a resolution of about 14 hertz in your C alpha dimension. And traditionally, today, we record spectra that are about 50 to 60 hertz in the C alpha dimension. So which means there is a lot of room for us to gain in resolution, which could be used to assign these large proteins. So if you believed what I told you and you go to your spectrometer and then you start recording HNCA on deuterated proteins for a very long acquisition time with the hope of getting resolution, you'll be rather disappointed. And that's because this is what you're going to see with the traditional sample if you record. So after a certain time period, even though your C alpha promises you that resolution, you will encounter a problem, which is the coupling to the C beta. And that coupling limits your resolution to 35 hertz, which is the C alpha C beta coupling. Now, can we somehow take care of the C alpha C beta coupling to get that very narrow line that we want? There are some tricks, very old tricks that are in the book. The first trick that will come to mind is a constant time. So if we could evolve the C alpha as some integral multiple of the 1/2j, then what will happen is that the coupling will get refocused. But for the type of resolution that we are thinking about, you probably need to record 3 or 4 constant times because you're looking at resolution of about single-digit hertz. Now, here is something that we record this on a constant time on a GB1 sample, which is literally about 6 kilodaltons. Now, here is 1 constant time. Here's the experiment with 2 constant time, and here's an experiment with 3 constant time. As you know, if you go from 1 to 2 to 3 constant time, you're increasing the number of FIDs. What I want you to show is that as the lines get narrower, even for a very small sample, your intensity decreases because your first point, you're starting with about 100 millisecond delay. So this would never work for even ubiquitin, so forget large proteins. The other strategy that's in the book is what we call the selective C beta decoupling. So you can use band selective pulses to decouple C beta, which is a topic that I'll come into in a later part of my talk. But the traditional pulses that are available in native variant, they have 2 problems. One is that they would affect the resonances that are at the edge of your C alpha, and these would be glycines. The second major problem is that they also cause Bloch-Seeger shifts. Essentially, it's a homonuclear decoupling, and they would affect the peaks, the position of the peaks that you're recording, and that's not a property that's not desired. And again, here you're listening on C alpha, and while you're encoding C alpha, you're decoupling C beta. A third approach that's in the books is what we call a virtual decoupling. If we know what the coupling between C alpha and C beta is, we can deconvolve it in a computational method. Here I again show you 3 different strips. Again, this is a strip from an HNC8, and you can see a pair of doublets. That's a C alpha C beta coupling. Now, if I deconvolve, assuming the coupling is about 35 hertz, that peak gets beautifully deconvolved into a singlet, whereas that peak is completely perturbed. Now, because the coupling here is not 35 hertz, the coupling here is 38 hertz. Now, if I deconvolve with a 38 hertz coupling, that gets deconvolved, whereas the other peak gets perturbed. So there is no single solution for this particular problem. So what we naively thought, naively being the operative word, is that can we remove the coupling to the C alpha from C beta biochemically? So what I mean here is that, can I make a protein where only C alpha is C13 labeled and C beta is C12 labeled? And you'll see why the naivety in our thinking came from. So normally proteins are made, deuterated proteins are made with C13 deuterated glucose, and we thought of using pyruvate as a carbon source instead of glucose. And pyruvate is a 3-carbon metabolite, and you can purchase this metabolite that's carbon labeled either at position 1, position 2, or position 3, or any combinations, 1, 2, 2, 3. Now, if you use this as a carbon source, the way the amino acids are synthesized from this dictate the labeling pattern. So for example, the alanine is synthesized by a simple aminotransferase. And now if I choose a carbon that's a pyruvate that's C13 labeled at position 2, I get my C alpha labeled and my C beta would be unlabeled. If I choose a pyruvate that is carbon 13 labeled at position 3, I'll get a protein where the alanine is C13 labeled at the C beta position, but not the C alpha position. This is the simple of the amino acids. The rest of them are a little bit more complicated, and they get synthesized using one of the 2 different cycles in the bacteria, which is the TCA cycle or the gluconeogenesis cycle. And there's a set of amino acid also gets synthesized by a direct method using fusion. Now, for each of the amino acid, the labeling pattern, depending upon where you choose the carbon to be in the pyruvate, depends on the cycle they're derived from. For example, those amino acids that are derived through the TCA cycle, but from alpha ketoglutarate, would get their C alpha from position number 3. For anything that gets derived from oxaloacetate, will get its C alpha from position number 2. And anything on the gluconeogenesis that also transfers to the aromatic will get the C alpha from position number 2. And for the things on the direct pathway, alanine, lysine, and valine would get the C alpha position number 2, whereas leucine will get it from position number 3. And actually, we should have done this before we started the project, but luckily we didn't do that. Because if we have done this, we would have probably stopped the project at this particular step. Now, there's a further complication that occurs as well, and that complication is that things that enter the TCA cycle, for example, pyruvate enters the TCA cycle as a 3-carbon metabolite, and then fuses with oxaloacetate to form citrate, which is a 6-carbon metabolite. Now, this fusion process, depending upon the pyruvate that you use, reintroduces the coupling because a carbon and a carbon can be right next to each other. So which means for some of the amino acids, the coupling will come back. Now, I actually want you to keep in mind 2 amino acids. One is glutamate, which gets its C alpha from pyruvate at position number 3, and alanine, which gets its C alpha from pyruvate at position number 2. Now, here are 3 different samples that we made of GB1. One made with a pyruvate that's labeled at 2 and 3, one made with pyruvate that's labeled at position number 2, and the other made with pyruvate that's labeled at position number 3. And here I show you a strip from an HNCA experiment. And you can see that once we have the pyruvate labeled at both positions, you get the C alpha C beta splitting. If you have pyruvate that's labeled at position number 2, the alanine is a singlet, whereas we observe a reduced intensity for the glutamate, which gets its position from position number 3. Now, if you use pyruvate that's labeled at position number 3, we see a rather interesting pattern. We see a singlet here in the middle that is devoid of the coupling, but we also see a doublet where the coupling is reintroduced because of how it goes into the TCA cycle. So from this, none of these are perfect solutions to what we are looking for. So we thought, how do we reduce the coupling information and also bring back the resolutions that we need? So essentially what we did was we thought of doing two different strategies. One, make two different samples, one with 2 labeled pyruvate and other with 3 carbon-13 labeled pyruvate, and mix them in an NMR tube. And that wasn't a very good idea. But the idea that really worked for us is that to grow the bacteria with an equal combination of 2 and 3 13C labeled pyruvate. The idea here is the following, is that when that recoupling happens in the TCA cycle, the chance of it introducing another coupling is less over here. So now we can see with this, what we call a pre-mix solution, where both the pyruvates are added and thereby the recoupling is statistically reduced. What you can see is that for alanine, you get a singlet, and for glutamate, we get this beautiful peak that is a singlet, but it also has a little shoulder that comes from the residual coupling. And what we realized post facto, which we didn't expect what we started off with, is that each of the amino acid has a central resonance for your C alpha that is devoid of any coupling, but it also has a shoulder, which is the coupling that is introduced by the biochemical pathway. And this shoulder is specific for every amino acid that we could think of. And the amount of this particular shoulder depends on the biochemical pathway. So to give this, put this into a slightly more clearer terminology, for alanine, we see the central peak that is devoid of any coupling. And for isoleucine, we see the central peak that is devoid of any coupling, but we also see the shoulder wherein the coupling is reintroduced by the biochemical pathway. So we can quantitate this by the ratio of the coupled to the uncoupled. And if you plot that ratio of the coupled to the uncoupled for different amino acids, anything that comes through the direct pathway or the gluconeogenesis pathway has a lower ratio, which means it has very little of the coupling that is reintroduced. And I think that's to the TCA cycle or the fusion pathway has slightly more of the coupling that is reintroduced. I want you to grab your attention to this number. It's about 50%, which means that's the maximum amount of coupling that will be reintroduced, which means your central peak that is devoid of the coupling is going to be twice as tall. So this method of labeling with pyruvate gives you two key information. One is a central peak that is devoid of coupling, which is extremely sharp, which we can use it to align. The other is what I like to call the residual scalar coupling, that is as a result of the labeling, which is amino acid specific, which is another factor that we'll also use in the resonance assignment. And here I show you examples of both. Now let's look at what the enhanced resolution, which is devoid of the coupling, can give you. Here I show you an HNCA again from GB1, which is the sample is made in glucose and recorded to a resolution of about 42 Hertz. You see one central peak over here. Now, when you take the same sample and increase the resolution, because the sample is deuterated, you can increase the resolution. What you see is that you see this particular pattern. And upon proper investigation, you know that there are two peaks over here. You can see one peak over here, and the sequential is actually rightly bordering right over here, that's shown in green. Actually, we get the sequential information from an HNCOCA. But it's very hard to see there are two peaks over here. But now once you collapse that using the mixed pyruvate sample, the coupling is collapsed. You can clearly see your internal and the sequential peak. And this is what the idea of what the resolution in the absence of coupling can give you. It allows you to clearly see the peaks that were previously overlapped because of either resolution or coupling. Now, the idea here is to assign, use this method to assign very large proteins. And our goal was to take MVP, which is a 42 kilodalton protein, and use a single HNCA experiment to assign MVP. Now here I show you the primary sequence of MVP, and where each amino acid is color-coded in a heat map by the number of matches they would have in an HNCA spectra. For example, if I take that tyrosine and ask and sit on the C alpha of the tyrosine and ask how many C alphas are matched for the C alpha -1, you would get close to about 20 different matches. And you can see that things that are in green are those amino acids that we can clearly assign with an HNCA that is recorded to a resolution of 42 Hertz in the C alpha dimension. So the take-home message from this picture is that for most of the amino acid, you have upwards of 15 matches for every single C alpha residue. Now, if you take the same sample and then you record it to a resolution of about 4.8 Hertz, which is what this labeling method allows you to do that, now the picture becomes very different. Now we can see we get a lot more unique matches that are shown in green. And now the matches, even for the things that we're not able to unambiguously assign, just using the chemical shift of the central peak, now drops from 20 to 2 to 5. So the maximum we have is about 5 in terms of degeneracy. Now we can address this degeneracy if we could also use the peak pattern. Here I show you an example. I'm sitting at the C alpha of a particular residue 259. I'm looking for a match. I get 2 matches that are identical in peak position, but only one of them matches the shape that I'm looking for because they have to come from the same atom and the same amino acid. And the other one, even though it has the same peak position, does not match the peak pattern. We simply calculate a correlation coefficient between the possible matches, and the one that has the highest correlation coefficient is the match for the assignment. Now, using both the information of the central peak as well as the peak shape, we're able to get about 95% of the MBP assigned using a single HNCA experiment. So that's the power of the labeling method, which nature gives us. So essentially, if you use pyruvate, what will happen is that you'll get a central peak that's devoid of any couplings, and you'll also get these residual couplings that are amino acid specific, which depends on the biochemical pathway, and you can use the combination of these two to assign them. Again, this is resolution that's beyond the Nyquist grid. Now, here again, I show you the peak shape, the reproducibility of the peak shapes. And here's the of valine, different instances of valine that you can see that. And you can see from the C to the coupled to uncoupled ratio, they look very, very similar. Some practical considerations that if you're going to use this method, you definitely need to have very long acquisition times for you to get that resolution. That's about 60 to 90 milliseconds. And deuteration is mandatory. Without deuteration, the C alpha relaxes way too faster for us to observe any of these things. It's very easy to deuterate pyruvate. You don't need to buy deuterated pyruvate. This is the only hydrogen that will that'll be there, and this hydrogen is very acidic. So essentially, all you need to do is that put it in a base, take the detour that you're going to grow, drop the pH to about 13, add the pyruvate, wait for 30 minutes, and then just add the phosphates to re-neutralize them. You get your deuteration. Now, here I showed you a way using a labeling strategy to get resolutions and peak shapes that are useful for assignments. So we take the simplest experiment, that's HNCA, and then we are able to add more resolution in a non-Nyquist fashion to get to where we want to go. Now, the question is that, can we achieve the same thing using RF pulses? So the idea here would be, can I take a sample that was made in using glucose and achieve the same thing here? So here's a problem that we are trying to solve here. So you take a regular HNCA experiment that is very sensitive. It's also good to note that in the HNCA experiment, the carbon where we're actually encoding this higher resolution is transverse only during the encoding time period. That's the other advantage of this experiment. If for students who are following the HNCA experiment, you have an init from H to N, then you have another init from N to Ca, then you flip the Ca back, you encode the Ca, then you go back to nitrogen, and then you encode the nitrogen in a constant time on your way back to proton. So which means you can spend all the relaxation that you have on the carbon into encoding the resolution on the carbon. But if you do that using a traditional sample, as we saw previously, you'll be limited by this C alpha, C beta splitting. Now, what if during the evolution of the carbon in the indirect dimension, I have a pulse that would faithfully invert the magnetization of the carbonyl and invert the magnetization of the C beta? Then essentially what I can do is that I can solve this problem. So we need a good pulse that will do that without causing any Bloch-Seeger shifts. So for this, we reached out to Paul, who's a student of Naveen Kaneja, and to use optimal control theory, or optimal control theory-based pulse design. Again, that's a field that was pioneered by Stefan Glasser and Naveen. And Instead of regular RF pulses, here you give the starting point and the ending point of the magnetization that you want to. If you want the magnetization to go from plus Z to minus Z, those are the starting and ending point, and you let the algorithm decide how the trajectory of the magnetization is going to follow. So here are some examples of some of these pulses would look like. So here's a 1 millisecond optimal control pulse, and that's the change in the RF, and that's the change in the phase during this 1 millisecond. It's also key to note that you only give the starting and the ending point of the magnetization. So magnetizations at different chemical shift would start at the same point and end at the same point, but they might follow different trajectories along the Brock sphere. It's almost philosophical. You start and end at the same point, but you have different routes. And so using this, we want to accomplish two things. We want to accomplish a decoupling, but also the decoupling should not have any Bloch-Ziegler shifts. So here I show you this pulse. This pulse is essentially offered during the C alpha encoding time period. And here you can see that. So essentially when you're encoding C alpha, we offer this pulse. And when the pulse is not there, the traditional setup, you see the splitting, the doublet. And you can see that when the pulse is offered, that doublet collapses into a perfect singlet. And that singlet is exactly in between, so which means we don't have any Bloch-Ziegler shifts. And you can see from the 1D trajectory, and I'll also show you later, that you get a twice increase in signal height. And the implementation is rather very simple. Again, if we look at the HNCA pulse sequence, you see again the inept from proton to nitrogen, nitrogen to carbon. And this is the traditional way that if you open up a Bruker pulse sequence, that your CA encoding is happening. So to prevent Bloch-Ziegler shifts from CO decoupling, you offer actually the CO pulse twice, which is sandwiched by a 180 on the observed resonance. So this essentially undoes the Bloch-Ziegler shift that is caused by the pulse. So essentially, this is how complicated your traditional CO evolution just to compensate for this Bloch-Ziegler shifts. We do something very simple here. So that's your carbon evolution time. And what you need to do is that you just need to refocus the nitrogen coupling because carbon is deuterated and you normally have a decoupling. So you just put a single 180-degree pulse for the first millisecond. Then our pulse that does both the CO and the C beta coupling comes after the first millisecond. You go to a different block of the encoding and you just put two pulses. It's very simple, right in the middle of your evolution that refocuses your coupling to the nitrogen as well as to C alpha and C beta. Now, when we compare to this, the C beta decoupling pulses, we generated 3 different versions of them, which have different bandwidths in which they can decouple. For example, the red one is called the BATCOP1, which decouples anywhere from 10 to about 30 ppm. The purple one decouples pretty much every amino acid. And if you can see that the red one does not decouple the amino acids that are leucine, aspartate, tyrosines, the ones over here. And the yellow decoupling pulse only decouples this amino acid. It does not decouple alanine nor this. Now you can clearly see that when we offer this, when we run a spectrum with this, you can see two lines, one with a slope of 1, another with a slope of 2. Essentially, you get the twice increase in signal height, which you'd normally not see with traditional pulses. Now, the other beautiful thing over here is that you also get different patterns. So for example, glycine doesn't have any C beta. It's a singlet in everything. Now, for example, let's take things like alanine. So alanine is a single with the purple with the purple pulse as well as the red pulse. Alanine C beta is at about 18 ppm. So the purple and the red pulse flip the C beta of alanine, whereas the yellow pulse does not flip it. So you'll see a doublet. And for something like aspartate, you can see again aspartate is over here, which is only covered by the purple pulse, and it's a doublet elsewhere. So now essentially what we're doing is that we're running 3 different HNCAs, and depending upon the amino acid type, and just by putting a single 180-degree pulse in the middle of the evolution, we get amino acid specific information. And this could be used to solve the same problem of degeneracy. Let's again look at the example that I showed before. I'm setting up the C alpha, I'm looking for matches for my C alpha -1, and I find 3 different matches. Now, if I follow the profile with these 3 different HNCAs, only one of them will follow, the other would not follow. So I know the peak that is right next to it is a match, and the others, as you can see, that's a singlet here, whereas it's a doublet over here. And that is literally related to the C beta chemical shift. So essentially, we are encoding some sort of a C beta chemical shift information in a gross way, but just running HNCA experiments. Now, the question is that, can we do better? Now, these pulses that we initially designed called the bad cops, they are binary pulses, which means they are either on or off. So for example, the red pulse is on in any bandwidth between 10 and 35, and after that, that's off. Now, what if we start creating, instead of binary, a gradient decoupling along the profile? So the idea is that, now the way to look at this, the graph is that 1 is where the magnetization is not flipped, and -1 is the magnetization is flipped. So when you flip the magnetization, that's how you get decoupling. Now, with that in mind, now let's look at, so this is the profile of what will happen along your C beta frequency while you're observing C alpha. Now, if a magnetization is flipped for a C alpha whose C beta frequency is at this particular point, you'll get a singlet. If the magnetization is not flipped, you'll get a doublet. And for anything in between, you'll get something in between, which is a combination of both doublet and singlet. Now, the pulses actually look a little bit more complicated here. We normally use about anywhere from 3 to 4 decoupling pulses. That is 3 to 4 HNCAs that you need to record. And here you can see 4 colors: blue, purple, yellow, and red. And here again, you can see what the decoupling profile is along the C beta frequency. Now, for a C alpha, whose C beta is about 38 ppm, the yellow pulse would flip it, and then that'll result in a decoupling, resulting in a singlet. The blue pulse would do nothing, and that'll get a doublet, and things in between would be a combination of singlet and doublet. Now, for the same HNCA experiments, if my C beta was at a different position, now the corresponding C alpha, remember here the blue was a doublet, and now here the blue becomes a singlet. And then the rest are doublet and singlet, depending upon where the pulse hits them. Now, this is only accounting for the triplet. Here, what you observe here is coming from either coupled or uncoupled to the C beta. And now we can do the same thing with the carbonyl as well, and we get this beautiful pattern where any one of those combinations can happen. The triplet is a quintet, essentially having coupling to both C alpha and CO and the combination of both. Now, if you think about this, this becomes a mathematical problem right now. So depending on where my C beta frequency is, I get a particular pattern in my C alpha, and I use this space of decoupling to faithfully encode all possible frequencies. So now the idea is that I can take these two information because I know what pattern I'm getting experimentally. I know what the coding frequency I'm using. Now I can not only use this to disambiguate and get specific peak shapes to help my assignment, I can actually triangulate like a GPS what my C alpha, what my C beta frequency is without ever putting C beta transfers. So essentially that's exactly what we did. So here we are able to, this is an overfitting problem. So we have more variables than what we need. And essentially all we need to do is that to look for the pattern and the peak, as well as combined with information of the decoupling that we use. And we can absolutely triangulate the chemical shift from both for CO and C beta. And here what you see is that this is the experimental chemical shift for GB1 for C beta and CO. And here's a triangulated chemical shift. And you can see they form perfectly in one line. Now, the question people ask, does it work for larger proteins other than GB1? So here is MBP. You can see that you can get beautiful peak shapes for different residues, even when CO coupling is encountered. The other question people ask is that, okay, MBP is not a real protein. Does it actually work for real life proteins? And we have several real life proteins that are pretty much starting to precipitate after a few hours. You put it into the magnet, and they do work in these proteins as well. But the thing to keep in mind is that we're not doing anything different. We're just taking a simple HNCA experiment that is very, very, very sensitive, and then you're just putting a single pulse on the that orchestrates the CO and the C beta magnetization right in the middle of that to encode information into the C alpha line shape to give us peak shapes that will let us not only do the assignment, but also to triangulate the frequencies of the passive spins. We call them belugas, and the reason I just love these mammals, but they also, the pulses are called C beta assignment by echolocation using gradient decoupling. And the belugas actually do work by echolocation as well. And so I think in this, I presented you two methods. One is using the biochemical labeling using pyruvate that gives you very sharp central peaks that's devoid of coupling, but also peak patterns that is dictated by the biochemical pathway. And I showed you another way of using optimal control pulses to derive the same information. But these also could be used synergistically because the information from one is independent of the information of the other. So hopefully using these methods, we'll be able to assign large proteins. And I think I should probably qualify what is large and large proteins in the era of cryo-EM. We think we can go to proteins that are about 250 kilodaltons, at least from the relaxation measurements that we could have. With that, I would like to end my talk thanking the people who did the work. The optimal control design was done by Paul. The pyruvate part of the work was done by Scott and Abhinav, and the pulse sequence development and the data for both the optimal control was done by Abhinav. And got a lot of help from other members in the lab making different samples to try this out. And we also got some help from Wolfgang for some of the fancy things we are trying to encode with the pulse sequence, which I haven't talked about. And we want to put these functionalities and modalities of matching them into a software that everybody uses. So we're working with Gersten on incorporating them into CCP NMR. And finally, thanks to Ilya for doing some of the simulations for us to tell how fast our magnetization would relax. So with that, I would like to stop, and I would like to be more than happy to take any questions.
Sulakshana? Hello, Sulakshana? Are you there?
Yeah. No, I actually forgot to unmute. Sorry.
Okay.
Thank you, Hari, for the very interesting talk. I think, okay. No, the first question I have is, see, you have used this technique, two different techniques. One is the pulse sequence method, and the other one is the using different labeling. So for large molecular weight proteins, have you used the same technique for assigning intrinsically disordered proteins?
No, we haven't done that. So as you know, we could. But as you know that my heart is in a different place, I'd like to use nitrogen detection for that. So we haven't used it, but there's theoretically nothing that stops. Actually, it should be even better because you'll get phenomenally better line shapes, and you also need not worry about back exchange, which is another problem in these steps. Yeah, you absolutely can use it.
Especially for those proteins which has a folded domain followed by a long intrinsically disordered tail. For those type of proteins, so it becomes huge also, and at the same time.
Yes.
So you have many questions now. So the first question is, does double S3E decoupling method can help to improve the resolution?
I would like a little bit more clarity on that question. Double S3, I thought the S3E is mostly on the nitrogen side back, and here we are talking about, so is that, I think it was Jay Subbareddy, are you talking about improving the resolution of the nitrogen dimension, or? Yeah, I know that's a spin state. It's a alternative to ST2PT, but that would work at least my understanding of that, it'll work on the nitrogen dimension. And here the problem is that we're also trying to minimize the time that we have for experiments. So we use non-uniform sampling, because if you would record, we record here about 1,024 points on the carbon dimension. And so essentially we like to limit the resolution of the nitrogen dimension to the constant time that it allows, and then put everything on the carbon dimension, because that's where we need the resolution. And we use about anywhere from 8 to 10% of the sampling of the grid using of the Nyquist grid using non-uniform sampling. So if you're referring to things on the nitrogen dimension, of course, yes, you could use that to improve the resolution of the nitrogen, but we here are seriously compromised for time as well. The ST3 works for CO-CA decoupling. But the CO decoupling perfectly works, right? Because CO decoupling, there's no problem. You're far away. You're not worried about any of the Bloch-Seeger shifts, and you can also decouple them by just putting 2 of the pulses. The interesting here is the C-beta decoupling. And the CO decoupling, the concept I which I talked here is that now you get CO decoupling that is actually band selective. So I can create these shapes on the CO And I don't think an S3E would work with it. S3E would probably give you a perfect CO decoupling. And here I'm not looking for a perfect decoupling. I'm looking for a decoupling that is CO chemical shift dependent.
How do you handle the serine and threonine residues with pulses?
Very badly. We don't. It's very close. The serine C alpha C beta is very close, and that's why if you look at my graphs, we don't. That's something that we have some ideas of how to do it, but it's still it's one of the harder problems. The serines and threonines, we don't handle them. We can't triangulate them.
How does this work along with peak picking and auto assignment?
The peak picking is pretty simple because you pick the central peak. The auto assignment, we have a home built that's a Python script that Scott wrote that calculates the correlation coefficient. But we are trying to implement this in CCP NMR, where you no longer look at the central position, you also look for the correlation of the peak shapes. And that should be hopefully available very soon to the community. We're still trial testing that. And we also want to involve some sort of a machine learning because now if you take the pyruvate method, the peak shapes are just amino acid dependent. And if you could make 20 different samples, and I'm working with Ilya Kuprov on this, and you can get some sort of a machine learning to a priori without even the information about the shapes or the ppm to see if we could triangulate this into a particular amino acid type and use that information as well. But these are all still work in progress.
How well does E. coli grow on pyruvate compared to glucose?
Not very well, but the yield is pretty much the same. The only problem you're having with pyruvate is that the growth is slower. The yield, if you wait for it, the yield will be pretty much the same, but it'll probably take about 1.5 to 2 times slower. And we have some tricks where we try to adapt it. So essentially, we grow the E. coli instead of directly going from a mini culture. We try to go into the pyruvate and then train it, and that'll. So the answer is in terms of yield, you'll see very little difference, but in terms of growth, you'll probably see some more time.
With this, we thank Dr. Hari for the wonderful talk. Thank you, Hari. And
Right, a pleasure.
yeah, and next week, our speakers are Dr. Mandar Deshmukh and Professor Murli Krishna. So we look forward to next week's talk. And so with that, we end today's meeting here.
So thank you, Sulakshana, for conducting this meeting today. Unfortunately, today what happened was during the talk of Neeraj, some person barged in and tried to play some mischief. We immediately removed him, but we'll anyway discuss how to avoid such things in future in next Zoom out talks. We are sorry about what happened, and some unknown thing came, and some love signal, heart signal, everything came on the screen. It is some mischief played by an unknown person. I'm sorry about it. Anyway, so we'll we'll correct it from next time onwards. And such mischief mongers we want to avoid and remove them permanently. Thank you very much for participating. We'll look forward to see you again next week. Thank you all.
Okay, see you all next week. Bye.